with the first talk, which is Helen Saibil, who's going to talk to us about the new national facility.
Okay, hello everyone. Hopefully we'll make up some time because I'm not going to go on for the schedule. Really, I have some stuff to present, but I think people probably have different questions about this. Some of you know a lot about it, some of you may not know anything about this national facility. So I'd rather just use this for questions, really. I'll give a brief presentation, then people can ask those burning questions that they have. So we decided to call it eBIC, as Electron Bioimaging Centre. So it was originally a Wellcome Trust strategic grant application by that group of people. Gerd Matterlich was the head of Dyna at that time. He's retired now, but he was very important in getting it all started with Owen Dave Stewart. And Kai has really done quite a lot of the work to get things going, because it's already almost going. The building, you may have seen, it's just, I don't know which direction, which way is it? Somewhere over there. It's behind that satellite dish thing, so you can have a look at it when you walk by. It's not ready on schedule, but it should be ready this year, we hope. So I'll just explain what the setup is for those of you who don't know. So the idea is that we want to have it basically like a beamline, but for cryo-EM, for state-of-the-art single particle analysis and cryo-electron tomography. And the idea is that people would apply with projects, they would prepare projects. So in general, the way it works is you have to have a lab where you have to have access to some basic cryo-EM so that you can get your sample worked out. You can't just show up with a test tube of protein or something that you have no idea how to make an EM grid of it. That won't really work. These are expensive microscopes whose time is still going to be very much in demand, and the projects have to be at least basically worked out to the point of making feasible grids. And so then on that basis, people apply for beam time as they would for an X-ray beam, and there's a peer review panel that would just allocate time. Part of that time will be at the Oxford Containment Facility, OPIC, at the Wellcome Trust Centre. What's it called? Wellcome Trust Centre in Headington, and that's ultimately, eventually, that will be for Category 3 for infectious things. But for the moment, because it's not all going in, there is a sort of startup program happening there. And so the idea is this would really run like a beamline, 24 hours a day, days when we're working. And so it has been funded by half by the Wellcome Trust and 1/4 each by the Medical Research Council and the BBSRC. So, as I said, the aims are to get a really well-working facility that's properly supported by experts and will be just run like a beamline. So I've probably said all that. There is a certain aspect, well, there's some extra aspects that we've designed into it. One of these is to have facilities for helping people get going. So the idea is to the extent that we'll have capacity, is to bring in either people from the crystallography side or from the cell biology side or other communities who don't know about cryo and who want to get started. We will have some facilities for sample preparation, and we'll try and have some kind of program of help and training, but that won't, obviously that can't be so comprehensive for everybody because there won't be enough resource, staff and time for that. But the idea is to help bring people in to the field, and also to have a strong in-house research program so that there's biological research going on using the facility at the same time, so things are maintained or developed to their with some sort of continuing development of the facility. And obviously we want to be very closely connected with CCP-EM. We're going to create quite a demand, we hope, with lots of sort of jumbo amounts of data flowing out of this thing, and with the EM database. So that's the general idea. So this is the management scheme as we originally planned it. It's not all in place yet, so we're still in the process of recruiting staff, but that's going okay now. We have various things happening, so I'm hoping perhaps in another month or two we'll be able to make some more specific announcements about that. So there'll be an advisor, a program board as the original people, I guess, who started it, plus some other representatives. The scientific advisory committee will be constituted from some external people. and also diamond people. And it'll just, all the mechanisms will follow the standard, what's already very standard for diamond. For those of you who know about applying for X-ray beam time, diamond beam time, it's all very well organized, and we'll just plug into all those same administrative systems. And the grant has given us 5 years, which is ticking away rapidly, with a core staff to run the thing, to keep it all going, and to pay buy all the equipment, pay all the user costs, and contribute to building a building, which is partly, so the building is partly the EM facility, and it's partly a new beam line, a new diamond beam line. So the grant really covers everything for 5 years. Our task is to demonstrate that it works in the 5 years, that it's a working user facility by the end of the 5 years with everything going. If it's not, we're in trouble. If it is, then the idea is that it will be continued. So we have to make it work. So I think, I don't know if I need to spend too much time on this. So I guess not everybody here is from the EM field. Many people are, but I can see some faces.
No, there's definitely some crystallographers.
Some crystallographers. Okay, so I'll just briefly say. All right, so I'll just briefly say that there's been steady technical advances in the EM field for quite some time, but they've suddenly come to everybody's attention because there've been these, spectacular set of high-resolution single particle structures, and suddenly all the crystallographers have thought, I can fix all my problems by doing that instead. It's not going to be quite that easy, but the direct electron detectors have made a huge difference because the previous CCP detectors were just not very good for this purpose. So things work a lot better now. And again, I think if people have specific questions, we can go into that. You see some experts here on that who can also help. So there are some other developments that might make a difference for some things, particularly phase plates. So for tomography, for getting low resolution contrast, it could be quite interesting to have the new sorts of phase plates, and we're going to try that. It's not obviously useful or necessary for high resolution single particle work, but it could be very interesting for some kinds of experiments. So the whole range of stuff, we've shown this in lots of context, the whole range of projects can be accommodated by this equipment, everything from the high resolution single particle or ordered arrays, probably microcrystal electron fraction if we get around to that. There's a demand, and different sorts of assemblies, and tomography of whole cells is probably one of the hardest things, whole cells and tissues. That's really difficult to get very good data because there's just so much stuff in cells and tissues, and you have to slice them up if they're too thick. But also tomography of isolated assemblies, things like reconstituted things on liposomes or other big sort of complicated assemblies, but that have been somehow reconstituted in vitro. There's quite a lot that can be done by tomography, those sorts of things too. So that's the sort of range of biological things we are thinking about. So the actual stuff has been bought or is being bought, and the building plans. So there are going to be 2 Titan Cryos microscopes. The first one is already here on the site. The building's not ready, so it's in the diamond ring, and it's working now. Just there'll be a 200 kilovolt, similar but 200 kilovolt machine as well. There is a focused ion beam milling machine, which the cryotransfer requires FEI to do something, which is difficult to get them to do. And there's obviously going to be all the various kinds of sample preparation. And the idea is to have a pipeline for cell biology of correlative fluorescence, cryofluorescence, and cryo-EM. And on the OPIC side, they already have a well-established lab that does containment, category 3 containment for on a flower. So the local environment, some of you know about this, some of you don't. There's obviously there are a lot of different labs on this site. So it's not, as you probably noticed, it's not terribly convenient to get to, but once you get here, there's quite a lot of stuff. It's quite well organized, so I'll just put up those different things. One thing I wanted to mention in a bit more detail, because it is really sort of closely related now to electron tomography, is this cryo X-ray, soft X-ray tomography, and that's now gone on the beam line. I don't know exactly the state, whether it's working. Is this here? Oh,
She's coming
should be
tomorrow.
here.
Okay, she'll say something about it. But I just thought, because we actually did a try on the offline system, I just wanted to show, because we thought it was really pretty nice. So this is an X-ray tomogram of a malaria infected red blood cell at a very late stage, just before the parasites are going to burst out. They've already bust out of their vacuole in there. And you see the parasites as a sort of cloud of things inside that swollen red cell. And you have to use humongous gold particles for this. For EM people, they're very gigantic gold particles. But what you can see, which is very nice, is the membranes. So for some miracle, even though the resolution is not very high, the membranes show up pretty well. So if you want to look at systems where you want to follow membranes in 3D without having to cut, this obviously goes through the whole cell. In EM, you can only get through a tiny slice of that cell at a time. So it's got the advantage you can see the whole intact object in cryo. So, and we see the software is very, very similar. It's to do these reconstructions. Tomogram images, this last year, which is a different one. Okay, so I think I'll just show a few photos now. So the building, this is actually out of date. So I think that scaffolding is all gone. So that's the buildings behind that satellite dish. You can see it up there. So we're supposed to be ready by now. Hopefully be ready later, towards the end of the year. The first Cryos was delivered a month or two back, and I'll just show a little. Yeah, so it's progressed beyond this stage now. That's a nice picture. So it all got put into the diamond ring. So there you see it's all sort of dumped in boxes there. Then the boxes were gradually unpacked, and it's actually running, and it's all assembled and running now in this spot there, and it seems to be running okay apparently. That's just one of the rooms in the new building, just to say that coming well. And this was 2 weeks ago. There was a beam, so there's a picture of the beam. So it's working. That's all we can say. So I think that's really all I wanted to present. I was more interested in people's questions. So, yeah.
So you say that it will be necessary to do some homework before actually coming and using this facility.
Yeah.
And you kind of mentioned Cryo-EM, but would it be okay, if you don't have cryo facility in house like we do in York? We have kind of standard old microscope, which is 120.
I'm not sure how we're going to have. That's tricky because it can be.
Because we can do negative stain, we can evaluate sample, of course, But would it be okay then to come and do some imaging here?
You'd have to get advice and perhaps work with people on site, or get access to a basic 120 kV cryo. But I think it might, sometimes it just works to go from negative stain to cryo, but often there's other things you have to do. So it needs some trial and error. You wouldn't want to go straight on a top machine without knowing that
But would it be also necessary then to prepare cryo samples in-house and then kind of evaluate them and then bring them over here? Is that the idea? Or?
Not necessarily. There will be grid preparation facilities in the lab. It depends. Obviously, for experienced users who know what they're doing, they make their grids in-house, they test them, they know they're okay. They can even ship them. And ultimately, it doesn't necessarily have to come. When we get started, I think it's going to be much more manual. But for people who are trying to get into the field, I think we will try, as far as we can, to give support to groups who don't have access to cryo. But that's not a routine way of using a high-end microscope. You have to get the grids.
Okay, right. And second question, so when it will be possible to get access, is it possible to get access now, or would it be kind of in
well, there is, okay, so the user program, for people who have, samples that are already worked out, they can apply for time on the Oxford instrument, and that has started. They're getting 20%. But it's going to start up gradually. We see that so the plan at the moment, this Cryos here that's in the ring, we hope to get that going. It won't be a sort of user program, but get it working and then leave it there until the building is ready and the second Cryos is delivered and installed in the new building, its final spot. Once the second one is working, then we'll move this one. So this is all going to take 6 to 12 months. It's not it's going to take we're talking about here time scales of half a year. Yeah.
How will the cryo sample transfer between the microscope and the beamline, the I-14 beamline, be accommodated? The beamline that is in the building is a beamline with the microscopes as well, but I thought the plan was that they would be able to look at the same sample, so to do correlative X-ray TEM studies.
You don't mean B24? You
No,
mean,
no, I mean I-14, the one that's in that building.
I don't know. I don't know how the sample, what the sample geometry is for that. I mean, can it look at really thin stuff?
I mean, it will be able to accommodate grids, but I hope it won't be necessary to unmount the grids from a Cryos sample holder into a diamond sample holder.
Well, I don't know. Mark, do you know?
I don't know a lot about this. I was under the impression that these were two completely separate facilities that happened to be sharing a building.
I wasn't aware that there was supposed to be a pipeline between them. It's not, certainly not biological. I mean, it could be done on material science. I forgot to say that the EM building also has provision for two high-end material science electron microscopes, so maybe that's...
No, I was under the impression that we would do nanoprobe fluorescence microscopy on biological samples.
Not that I know about. No, we haven't planned.
So it hasn't been talked about.
No.
You mentioned sort of applying for time on the microscope. What blocks of time are you considering? So for synchrotron resources, you don't need a lot of time to collect lots of data.
Yeah, no, you need more time. So it's probably going to be units at a day, I guess. Rather than a unit, because you can't, once you get a cryo sample on, it's not such a quick method.
Yes, I know that.
Yeah.
Well, you say that you will give support to people that are not expert in the field.
To the extent that we can, yeah.
Does this mean that you will do 3D reconstruction for a standard user?
Well, okay. So then the CCP-EM. So this is where CCP-EM comes in, because the sort of pipeline of what the aim is that there'll be we'll try and automate the data pipeline as much as possible so that people get feedback while they're recording data. They might get preliminary tomograms or preliminary class averages if things really work. So, if we can get all that working, then that would already give some feedback to the users. But for users who don't know how to do any processing, I think that's a more general community training question. There will be training courses. It may not. I don't think each user will be trained. Well, we won't be able to train each external user individually, but there'll be some kind of program of training, and hopefully that will be combined with CCP.
Yeah, I mean, CCP-EM's goals would be to install automated pipelines that, as Helen says, will give you quick feedback. But I don't think we're going to do a kind of a full user service, and I don't think that would work.
Well, not in the first instance, for sure.
Yeah. Well, I mean, I think ultimately it's the researcher's responsibility to understand their own structure and to do the final refinement and validation. I mean, that's how it works on the X-ray beamline crystallography. It's not something that beamline people would take responsibility for. So
it's not feasible to look after everybody.
I mean, because every problem is slightly different. Every structure is slightly different. So I don't think that would be feasible.
Yeah.
Can I just comment on that as a crystallographer whose group are learning EM just at this point? Rely on software is actually beautifully written, as is the documentation. So we have collaboration with UPAR and OPIC, and with his help we're working our way through solving our first structure. It's not totally impossible for crystallographers, with some help and supervision to make sure we're not making horrendous mistakes, to actually use that software now and hopefully get towards a structure without having to have somebody do the whole lot for you. We hope, anyway. We're only halfway through.
Yep.
Is it also possible for labs outside of the UK to apply for it?
Sorry, outside of the UK. I think to the extent that Diamond has international users, I think it's also possible. Yes, I think the main criterion for projects coming in should be scientific excellence, and that should be flexible enough to.
What detectors were planned to go on the Titan Cryoses? You said direct electron detectors. What?
Oh, which ones?
Yeah.
Okay. So we've chosen the Catan K2 for the first one. That's turned out to be, that's quite a problematic choice because FBI, the microscope manufacturer, doesn't cooperate properly with Catan, and they're, although they agreed to make it compatible with the FEI software, they have been dragging their feet in excessively, and so there's technical and commercial problems to do with that, I'd say, but we're going to insist. Yeah. So I think they've given us their detector. They're trying to basically make us use their detector.
So it would be the same on both microscopes?
Not necessarily. We're still looking at, that's a developing field, and we're still waiting to see what's going to happen, what the next generation's going to look like before we make our next choice. That's something which we hope to stay, up to date with, or at least be able to keep updating at some sort of time intervals.
Okay, if there are no more questions, should we thank Helen and move on to No, there are ceiling, yeah, it should be picked up by those. So let me introduce the next speaker, John Briggs from Heidelberg. So take it away.
So thanks for the invitation. It's nice to be here. Am I talking loud enough for those of you at the back? Yeah. So in my lab we're interested in the. Thank you very much. I think there's enough beams in this place that. Okay. So my lab is interested in enveloped viruses and coated vesicles. So we work on HIV, influenza, Ebola on the virus side of things, and then we work on COP1 and Clathra and coated vesicles mostly on the coated vesicle side. So these are machinery that are assembling onto some piece of membrane in the cell and are bending that membrane. At the same time, they're collecting a defined cargo, which is either the proteins in the genome and the accessory factors of the virus, or are the cargoes of the transport vesicle. So they're collecting those things together in one place, they're bending the membrane, they're going to form a bud, and they're going to pinch off to form a vesicle, or a virus. And this is then going to identify and fuse with a target membrane to deposit its contents into the new system. Okay. How the projector knows that I'm not allowed to use that button. So if we want to understand how this works, and to do that we want to understand the structures of the proteins that are involved in situ, so on membranes and ideally during budding events. We want to see how those change over time, but this is a very dynamic process. So this brings us to the question in situ or in vitro, and this is the kind of structural biology spectrum. If we work with very simple systems with highly purified proteins, we might be able to crystallize them, we might be able to do single particle EM, and we can generate very high resolution structural detail. If we want to look at more complex and more variable systems, or if we want to look at components in situ, perhaps even within cells, then we're limited to obtaining low resolution information. So depending upon what questions we want to answer, that will affect what level of detail we require, and which will affect where on the spectrum we need to do our experiments. So I'm going to very briefly give an example from the virus side of things, and then mostly I'm going to talk about coated vesicles, but if anybody wants to ask me questions about the virus stuff later, happy to answer them. So this is the assembly and budding of a virus like HIV, and this is the same thing in a bit more detail. So in the case of HIV, the main structural player is a polyprotein called GAG. It has a series of folded domains collected by flexible linkers. And this protein, when expressed, you can express it just by itself in a cell, or during an infection, will self-assemble, so it interacts with itself via CA-CA interactions, so capsid-capsid interactions. It interacts with the genome via the nucleocapsid domain, and with the membrane via the matrix domain. It assembles these layers which curve the membrane and bud out to form what we call the immature virus particle. So this form of HIV is not infectious. In order to become infectious, the viral protease is activated and cleaves the GAG protein at 5 different places, and that cleavage leads to a rearrangement of the virus structure to form the mature and infectious form of the virus. So we're interested in various aspects of this, but I'm going to talk about the immature virus. We want to know the structure of the immature virus in order to see the interactions between proteins that are mediating virus assembly, and in order to see the substrate of the proteolytic cleavage. So this is a cryo-EM image of immature HIV. You can make immature HIV by producing virus in the presence of a protease inhibitor. And you can see from such an image that each one of these virus particles is a little bit different to the one next to it. So the first thing we can conclude from such an image is that we're not going to be able to do a single particle reconstruction on the level of the virus. We can't average one virus with the one next to it, as you can do for regular icosahedral viruses. Nevertheless, within this virus there are protein-protein interactions and arrangements of proteins that are presumably the same in all of these virus particles. So there are repeating features, and they're within this protein lattice here, for example. So just to give you a guideline, this is the lipid bilayer on the outside, this is the capsid lattice, this kind of stripy layer underneath it here. We also can't apply 2D alignment methods even to the repeating features in the protein lattice, because this is a projection image, and the image of essentially one repeating chunk is confused by all of the density above and below it in the ice, which is being projected into the same place in the image. So in order to get structural data by averaging of the repeating features in such a virus, we apply subtomogram averaging. I realize I've aimed this rather at EM people, so I've skipped a step, but for the crystallographers that means we're starting off by doing tomography, by which I mean we place the sample in the microscope, we take an image, we physically rotate the sample and take another image, physically rotate it, take another image, and so on. So we generate a whole series of images of exactly the same sample from different directions. So from that series of images we can reconstruct a 3D reconstruction of that region of our sample, and that's what we call a tomogram. So this is a section of a tomogram, so we can imagine we've reconstructed a field of view like the one I just showed you, which contains multiple viruses, and we now have a 3D reconstruction of that. It's a low resolution in the sense that there's not been averaging done. We've applied the maximum allowable dose essentially to that region of the sample. So we have a low resolution 3D structure which has a missing information in Z because we can't physically rotate through 90 degrees, so the resolution in this direction is also lower. So here is a section, one virus, sliced through the middle. So again, you see the lipid bilayer and the protein layer underneath. So now we can extract what we call subtomograms, so small boxes containing parts of this repeating unit, and we can align and average them in 3 dimensions. So this is a kind of simplified scheme here. We have all of our subtomograms. They're randomly oriented. They're essentially picked from within the sample. We need to have some kind of reference. We align all of these subtomograms against that reference, so they're all in the same orientation, and we average them together. By averaging them together, we reinforce the signal but not the noise, and we're able to interpret higher resolution details. And we can use this average as a reference for a further round of alignment, and we can cycle around this loop in order to generate a 3D structure of the repeating unit within our sample. In this case, the lattice of a virus. So this is then one such 3D structure. So this is taken, this shows it to scale on one of the viruses. This is the membrane, this is the bilayer, this is the capsid protein layer, and this is the ribonuclear protein. This is a more recent structure done using the same methods but at higher resolution. So you can see this, these domains are these domains here, and in yellow are these domains here. So in this way we can get down to resolutions which are, in this case this is somewhere around 9 angstroms. At that resolution we can see the alpha helices, you can see them here as sausage-like densities. Because crystal structures of the component domains are available, we can then place those crystal structures into the map very cleanly and obtain a structure. So this is CCD data, and with direct detectors, with the greatly improved performance, it's possible to reach higher resolution and also to access a wider range of samples that might be more challenging due to flexibility or thickness or other such problems. And this approach, this subtomogram averaging approach, has been applied to a number of different samples by different labs. This slide's a little bit, a couple of years old already now. But these are essentially samples where one can't apply a conventional single particle approach because we're looking at the repeating object of interest in the context of a whole load of other machinery. So the rest of the virus, in this case, is the nucleopore from the rest of the nucleus, and so on. So I think, in principle, this approach is applicable to bits of cells, and I think this is where a number of labs are going. If one has a bit of a cell that's been prepared in the appropriate way, at the moment the state of the art is by ion beam milling, then if it's possible to identify repeating features out of such a 3D reconstruction, for example, the ribosomes, then one can average those together and generate 3D structures in situ. Okay. So this is just a slide for the hardcore EM guys among you, just to say that in the world of subtomogram averaging, there's a lot of room for improvement. If you think about doing a tomographic data collection, you're placing your sample in the microscope, you're going to take an image, rotate it, take an image, rotate it, take an image, rotate it. So you can decide your dose, your defocus, your tilt angle, essentially independently for all of the images in your data set, if you wanted to. Which means that there's a huge range of possible parameters in how you collect your data. And there's a lot of room for optimization in how you do that, depending on your sample. When you generate your final structure, I showed you in a simplified scheme that we're basically averaging together these 3D blocks. One can generate the final structure from the raw tilt data by calculating back all of the rotations and shifts. One can use different fractions of the data for the alignment and for generating the final structure. So some of these ideas are a bit similar to movie mode processing in single particle, where you might take a very long movie, applying a lot of electron dose, use the movie for alignment, but then only do your reconstruction from the early frames, which have a lower dose. One can apply similar ideas in subtomogram averaging as well. And there's no single optimal solution. The appropriate combination of data collection conditions and reconstruction approaches depends upon with the equipment you have, but it also depends on what the sample is and the properties of the sample. So this method will go to higher resolution than it currently is. Okay, I'm going to give an example of applying subtomogram averaging actually at lower resolution to what I've just shown you, but a sample that's more difficult to work with. And this is encoded vesicle budding. So if we imagine what happens, this is in a bit more detail. We have a membrane. It can be a plasma membrane or some organelle membrane. There are cargo molecules, and there's a cargo receptor, which, when cargo is bound, recruits an adapter. protein, which is this pale green protein, which then recruits a coat protein, and this leads to formation of a coated vesicle, which is then uncoated and will transport this cargo to some other membrane. And as those of you that come from biology rather than physics will remember from your textbooks, different protein coats are involved in different transport pathways within the cell. And I'm going to talk about COP1, which is involved in retrograde transport through the Golgi. So one approach that's been very successful in studying coated vesicles is to take the outer coat components and to express and purify them and to assemble them in vitro. If you take these proteins and assemble them in vitro, this is in the case of calafrin, you can form these regular cages, and these cages are an appropriate target for single particle reconstruction. There have been some very beautiful structures. This is the calafrin cage solved by single particle methods. So in the case of COP1, this approach doesn't work. So no one has successfully taken the outer coat components and been able to assemble them in vitro to form anything that looks like a coat. So instead we've been doing tomography. This is a tomography, Helen mentioned this is a tomography of in vitro reconstitutions. In this case we're taking giant unilamellar vesicles and adding in all of the purified protein components to get budding in vitro. And essentially the preparation really is as simple as that. We take the GEVs, we add in all of the components, and then we plunge it. And that's the preparation. There's no purification or anything afterwards. So you go from these huge vesicles, which are about the width of the room, down to these little coated vesicles, which bud off it. So this is the tomograms. These are all of these different views from different angles, and with gold used as fiducials for reconstruction in 3D. So from that set of tilted images, we can reconstruct that region in 3D, and this is a slice through it, and we can extract the individual vesicles, and you see, as for the virus, they're all a little bit different in size and shape, and we can't apply a single particle approach on the scale of the individual vesicles. This appears really to be a uniform distribution. So we apply subtomogram averaging, as I explained for the virus. We're going to go round this reconstruction loop. So what I'm going to show you is the initial average, when you just take all of your subtomograms and average them, and then we're going to align them against a reference and then average them. I'll show you the new average, and then we use that reference for another round of alignment, and so on and so forth. We're going round that alignment loop, and we do this twice for biological replicates. So you can see that over time the structures evolve and eventually stabilize. This is coloured then in the membrane in red and the proteins in blue. And if we compare them from the two experiments, we get the same structure. And we can see that we have this threefold arrangement of protein density, and that's the repeating element of the coat. That's this threefold structure. So we get another piece of information from the subtomogram averaging, which is where were those objects found. And we've had to calculate shifts and rotations in order to average these things together. So we know where all of the objects were, and we can visualize that. So if we visualize the threefold of the triangle, we can see for this vesicle, this is where those triangles were found. So we get information on the relative arrangement of the proteins in the sample. Okay, we'll go into the details. This is a higher resolution structure. This is current work from Svetlana. This is a structure at around 13 angstrom resolution. So in red is the membrane, and these are the proteins. And then I'm now showing the model, and I'll explain how we get there. And these are the various protein components of the coat. And within such a model, we can then see how they are arranged relative to one another and relative to the membrane. So I'm not going to let the movie play to the end. Oh. Okay. So this is then a representation of one vesicle with all of these, with the electron density. This is a subset of these are the protein components which ought to be present within the assembled coat. We know what proteins are in there because this is an in vitro reconstitution, so we put them in. And for some of them there are crystal structures available, for example beta prime. For others, there are homology models, and in many cases, or rather in the case of alpha and beta prime, in the case of these two subcomplexes, there are different but structurally homologous structures which appear rather similar in shape. So at a resolution of 13 angstroms, we're not resolving alpha helices, for example, and working out how to put these together into such a structure is non-trivial. Now, in some cases, the proteins have very characteristic shapes, which you really identify very clearly by eye. The way we've done this is to use Chimera, essentially, as a fitting program, and place the protein structure randomly in 10,000 different orientations and positions into the map, and allow it to fit and calculate the cross correlation. So that's the simple approach. And then to plot the histogram of that cross correlation, and then to look at outliers which have significantly better fits than the rest of the data. So in the case of fitting beta prime, we have these two apparently outlying fits. One is this fit and one is this fit. We expect to see two fits because alpha is a structural homologue of beta. So if we search with beta, we ought to also find out where alpha is. So we have two possible positions. And again, if we fit the gamma zeta subcomplex, again we get these outliers in the cross-correlation histogram, which are these two positions. So that approach can be taken with the various proteins that form the structure, but then it's then combined with labeling experiments where particular protein tags can be labeled, the structure can be solved in the presence of the label, and then the difference between the two structures looked at. That was how we positioned the alpha C terminus, the alpha C terminus and the beta N terminus. And then the final approach we took was cross-linking mass spectrometry, where a cross-linker is added to the system, which will cross-link lysines when they're within a certain distance of one another. So the cross-linker is added, the sample is then digested, and peptides are detected which are cross-linked, and when they're identified, it tells us that the two cross-links, or the lysines in those two peptides, are within a distance, within a defined distance within the map. And in yellow, you can see the positions of all of the detected cross-links. So based on the modelling and the labelling experiments, you end up with more than one possible way of putting the proteins together, but using the cross-linking mass spectrometry, you end up with a set of cross-links that is consistent with one of those models. So in that way, we generate a model of the COP1 coat. And it's at this level, one wouldn't want to interpret certain interactions between side chains and things between proteins, because we're talking about fitting into a map at 13 angstroms, and indeed in some cases fitting on the homology models. But the general arrangement of the proteins and the architecture of the overall coat can be interpreted. So I'm going to start off by explaining what we expected to see. So if you look at the proteins that make up the COP1 coat, one subset of them, what we call the adapter subcomplex, is a structural homologue of the AP adapters from the Clapeyron system, and that structural homology is very clear. The outer part of the coat, which is beta prime and alpha, so beta prime and part of alpha has been crystallized, and it crystallizes to form this very nice triskelion structure. And if you work on coated vesicles, you see a triskelion structure, and you think of Clapeyron, which also has this beautiful triskelion structure. And here we have a dimeric interaction between alpha and beta prime COP, and we know that alpha is a structural homologue, so we can assume that it goes out here and will also form a triskelion. And based on that, one can generate a beautiful cage model for the coat. So, based on these two observations, the assumption would be that this would look like a coat. So we'd have an outer cage, in the sense one has a Clapeyron, and we'd have adapters. Again, we have the APs, which are structural homologues of the Clapeyron system, which would link that coat through to the membrane. If we look at the structure that we see, so this is now the threefold basic building block of the COP1 coat taken out, in a number of ways it doesn't fit well with that prediction. So firstly, this triskelion that was crystallized doesn't appear inside the assembled coat. It appears to be an extremely elegant crystallization artifact. And the actual assembly of beta and alpha is an arch-like structure that links to the membrane on both ends. So there doesn't seem to be a triskelion structure. And indeed, there doesn't seem to be any real assembled cage of the outer coat at all, in the sense that the outer coat, which is coloured blue, and the adapter subunits, which are coloured green, you can see are in a very kind of interwoven interaction, where both of them need to be present in order to build this fully assembled structure. If we look at the architecture of the adapter and of the coat, they're interestingly similar. They both have membrane binding domains on the two ends. In one case it's ARF, and in the other case it's the cargo binding beta propellers. And these domains here are in both cases extended alpha solenoid-like structures. So the adapter and the coat are in very similar arrangements within the coat. And indeed there are cargo binding sites in both subcomplexes. So at least in COP1, although when you look at what's known about coats, and when you look at the proteins, you think this is going to be an adapter and a coat. When you actually look at the assembled coat, it's something very different, and we don't seem to have a functional subdivision of the two subcomplexes at all. So this is a kind of schematic of the two coats. In the case of COP1, you have this kind of interwoven mesh of adapter and coat, and the linkages between these threefold structures are made by flexibly attached domains. And the lower is the Blackman example. So the coats have a very conserved architecture, but they in fact polymerize in very different ways. We're thinking about coats now as things that distribute binding sites on the membrane rather than regulate curvature themselves through polymerization. And I guess the final message is that looking in situ is important. So you can, in this case, you can crystallize the proteins, but looking at how they assemble into this complex structure is essential to understand how they work. So that's an in vitro system. What we would like to be able to do, and which we hope to do in the future, is to address these structures in more complex environments, i.e. within cells. So to be able to say, okay, let's ask what does the COP1 vesicle coat look like, and then be able to answer that question by identifying the vesicles within cells and solving the structure at least at some intermediate resolution from within cells. And the challenge here is in many cases is in finding the object that you want. So I'm going to talk about classical-mediated endocytosis. These are yeast cells, 3 of them. Well, this one is budding here, and there's these are endocytic sites, these fluorescent spots. So there's a GFP signal being attached to one of the endocytic proteins, and an RFP signal being attached to another of the endocytic proteins. So what you see happening is these proteins are assembling at a site on the membrane, and when they do, you start to see this green spot as the fluorophores are collected together. The green proteins are arriving first, and then at a later stage, the red protein arrives, and when it does, the structure moves into the cell, and that's the process of invagination and pinching off. And if you were to watch this movie for long enough and to watch enough of such movies, you see that this process is extremely repeatable. So essentially every time you see an endocytic site, you'll see the appearance of a green spot, the appearance of a red spot, movement into the cell, and then it disappears. So in order to be able to identify these intermediate structures by EM, we need to be able to find them. And we're doing this using correlative microscopy methods. So what you see here is this is an EM grid. So these black bars are the copper bars of an EM grid. This is a sample that's been resin embedded. This is not cryo-EM. This is more conventional electron microscopy, where the cell has been embedded into resin and then sectioned. And this is a pellet of yeast cells that's already been sectioned. It's a section of 200 or 300 nanometers thick, and it's been placed on an EM grid. And then it's been placed in the fluorescence microscope here, and you can see the fluorescent spots. These are the endocytic sites. And this has just been frozen. The starting point for this preparation is a high-pressure freezing. So the cells are taken, they're high-pressure frozen, and then they go through the preparation method. So what you see here as fluorescent spots are places where endocytosis was happening at the moment the cells were frozen. So it's not synchronized or timed in any way, it's just a random step. So we can now identify a cell of interest, zoom in on that cell and identify a spot of interest, and then by correlating this fluorescence image with the EM image, we can locate the same spot in the electron microscope and within a tomogram. This is a 3D reconstruction of that region, and in green is the coordinate of the fluorescent spot, and that's the endocytic site. So you can if you have the fluorescence data, you can really identify what's a very transient intermediate in the endocytic process, and it's an intermediate which you would, if you were just searching, you'd spend a very, very long time looking for, and you wouldn't be sure that you'd found it, because if you find such an invagination, this could be an isosome or some other kind of membrane invagination. To give you an idea of the scale, the pixel in the fluorescence microscope is of this sort of scale, so it's a very different scale of information. Okay. Just excuse me a second. I think I've got them. Okay. So that what I just showed you was in yeast, and in yeast we've looked at a whole series of different intermediate states labelled with different combinations of fluorescent proteins, and have built quite a detailed model of the membrane shape changes that happen during endocytosis. What I'm now going to show you is mammalian cells, and here the question we had was a rather simpler one, which is there are two, and I think it's fair to say competing models, and I say competing models because, as you know, a lot of this depends upon the personalities who are defending one or other model at meetings, for how endocytosis happens in mammalian cells. So in one would imagine that clathrin first of all assembles to form a patch on the membrane, and that patch then bends to form a bud, which releases then eventually a vesicle. The alternative model is that clathrin starts to assemble on the membrane, and in doing so it bends the membrane, and it grows with the bud during endocytosis, and these models have very different properties. In the first one, the surface area covered by clathrin is constant throughout the entire process. It's essentially defined prior to membrane bending, and clathrin is changing curvature physically during endocytosis. In the second model, the surface area being covered by clathrin is increasing during the invagination process, but the curvature of that clathrin shell is the same at all stages. So we've used the correlative approach to address this question. We've looked at genome edited cells from David Rubin's lab. So these have clathrin light chain labeled with RFP and dynamin labeled with GFP for one data set, and another data set with clathrin labeled RFP and a cargo transferrin labeled with Alexa 488, also a green signal. And what you see on the left is are three different color channels in the fluorescence microscope image of the EM sample. So on the top, so I should say, this channel shows you tetraspects. These are small plastic beads, which are fluorescent and which are visible in the electron microscope, and they provide a coordinate system which is constant in the fluorescence image and in the electron microscope image, which allows us to calculate the image transform between the two extremely precisely. So these are those fiducials, and they are fluorescent in all color channels. That's important because you need to rule out chromatic aberrations in doing these very high precision correlations. But you see in the green channel we see a single spot marked with a yellow arrow, and in the red channel we see two spots. The top one is the same one marked with the yellow arrow. So there are two clathrin spots, one of which have dynamin in this case. And these are those two spots in the electron microscope. And this is then a tomogram. So you can see it's a junction between two cells, and these two endocytic sites are budding off into opposing cells. And again, this is resin embedded sample, but you get a very nice and clean image of the clathrin coated pits. And in some cases you can see the clathrin lattice. So when we collect this data, we do this for, in this case, a couple of hundred different sites, and the data is parameterized. So someone manually marks the membrane and the coat, and those are extracted then as coordinates and then treated computationally afterwards. So in both of the two models, the angle between the plasma membrane and the steepest angle of the invagination increases from the beginning to the end. So regardless of how you imagine this process happens, that angle must increase through the process. So we're simply using that angle as a surrogate for the stage of endocytosis. So when we look at our endocytic site, we measure the angle, and then we can in that way position it to be an early site or a late site. And then for each one of the endocytic sites in our data set, we measure the membrane curvature and the surface area covered by clathrin. And in fact, we can measure other parameters as well, but these are the two relevant ones here. So on the left, on the y-axis is radius of curvature. So the lower the radius of curvature, the more the curvature. This is the angles. This is the beginning of endocytosis to the end of endocytosis. And this grey range is the radius of curvature of the vesicles, the results. So this has to end up in this grey range, and you can see it does so. The radius of curvature gradually decreases and eventually stabilizes into this region. So the curvature of the membrane is increasing during the endocytic process. This is the point at which Dynamin is recruited, which is about 90 degrees. This means actually the Dynamin is already being recruited to very wide necks. And then this is surface area. Again, this is the range seen in the vesicles, and this is the range seen throughout endocytosis. It's a big range. This is because it goes in the square of the radius, and essentially it's not changing throughout endocytosis. And these are just a few examples of endocytic sites picked out of the data set. So we find the surface area is unchanged throughout the endocytic process. The curvature is increasing throughout this endocytic process. So we can decide that the top model is the one that explains what we observe in the cells, and the bottom model is not. This model implies that clathrin is playing a role relatively early in the process in providing a platform for recruiting cargo and for selecting membrane area prior to bending, as opposed to a model where by assembling into a rigid coat it's forcing a curvature onto the membrane. And this actually links the COP1 and the clathrin ideas, in that in both cases the vesicle coat proteins seem to be playing a role in organising the underlying membrane binding sites. Okay, then I've just got a few slides at the end about Cryo-CLEM. So what I just showed you was a sample that had been embedded in plastic, but of course what we want to be able to do is do the correlative approach on a Cryo-EM sample, so that we can really extract structural data from the sites which we find, and essentially do subtomogram imaging. So in order to do that, we need to do this correlation on cryo samples. That means we need to do cryofluorescence microscopy, and there are actually other reasons for wanting to do cryofluorescence microscopy independent of the correlation with electron microscopy. There are various solutions available. There's one from FBI, which was developed at the Baumeister lab. There's one from Lincoln, which was developed from the Costa lab. And I'm going to unashamedly talk about the one which we developed together with Leica, which is this system. And essentially what this consists of is a stage in the middle where your sample is held at liquid nitrogen temperatures throughout the time that you do your cryofluorescence microscopy. So this is a nitrogen dewar on the left. Nitrogen is pumped into the stage. This is the transfer station, which is the computer system. So inside the stage, this is where the sample is held. This is the access point. The reason for developing another system rather than use one of the existing ones was primarily that we wanted to try and improve the objectives that we were able to use in the system, which meant using a low working distance, high NA objective, and also to improve the transfer approach. So this system works with a 0.9 NA objective, which comes within 0.28 millimeters of the sample, but doesn't warm the sample up. And then the transfer system, like those of you that do side entry cryo-EM are familiar with this kind of thing. This is perhaps a bit more comparable to the Polaris system, in that you put the grid in, you transfer it into the microscope, you remove the transfer station, the grid is in the microscope. And then when you finish your FM imaging, you go back in and remove the grid, and you can then go and take that grid and put it into the electron microscope without the grid ever being exposed to air or warming up. And then the other thing which we like about the system is that it does full grid scans, including Z. So that means that you can place your sample into the microscope, and a couple of hours later you have such an image. There's an image of your grid in fluorescence. So you could look around this grid and identify the things you wanted to look at, and this image is a raster of much more detailed images, so you can zoom in and keep zooming in and really see your fluorescent spots of interest. So these are liposomes in ice. These are vitreous sections on a grid. And that's just, again, zooming in on just to give you an idea of the scale. And these are cells with, which have been transfected with 2 different colours. And again, we can zoom in on the cell of interest and identify the region of interest. And then the image data from the fluorescence microscope you take with you to the electron microscope, so that when you generate your grid map in the electron microscope, the 2 sets of images can be superimposed within serial EM, which means you can then click on fluorescent spots, and the EM will go to those fluorescent spots and take images. And the real advantage of this is that you can then look over your entire grid for very rare events and find them and image them efficiently in the electron microscope. That's the theory, and over the next few months we'll now try and use it to solve some structures that we weren't able to do before. Okay, so I'm just going to thank the people involved. So the COP1 work, the higher resolution structure was done by Svetlana. The project was started by Marco, and it's a collaboration with Felix Wieland and his lab in Heidelberg, and Martin Beck's lab helped us with the cross-linking. The high-resolution HIV subtomogram stuff I showed you was done by Florian and Vim in the lab. It's a collaboration with Hans-Georg Kreuslich. The plastic CLEM I showed you was done by Ori, and that project was started by Wanda and Martin, and that's a close collaboration with Marco Kaxen's lab at EMBL. And then the Cryo-CLEM is a collaboration with Leica, and the person in my lab who really made this work was Martin Short. And thanks for your attention.
We have time for a couple of questions.
I have a question. So you say that with the subtomogram averaging you can improve further from like 8 or 9 angstroms. So where do you place the limit of the actual reconstruction error, and how do you overcome that limit?
So in terms of how far you can go in resolution, then I mean, the limitations are similar to those of single particle, with the caveat that you're working with typically thicker, less regular samples where you have more things superimposed. But I think in terms of the image, in terms of the kind of imaging and reconstruction, then I think there's a fundamentally different limit there. Did that answer your question? No,
it's fine.
Yeah, it's like you're not going to go to
You have more dose, though.
Yeah, but you have more dose, but in the end you have a movie. So to some extent you can divide your dose how you like over your tilt series. You can use that dose to do your alignment, but you don't have to reconstruct with that dose.
But I mean, what I'm concerned about is like all the objects you are extracting are in different area of your tomogram, and you're doing like one rigid reconstruction of the whole tomogram. And I expect that you should be able to do some local refinement of the tilts of every box that you extract, if that makes sense. That was my question.
You can so one approach that other people have done is to take tomograms to generate to do a subtomogram reconstruction, and then to go back and refine the individual tilts. Now that approach works very nicely if your test sample was a single particle to start with, because then you have a clean image on which to align on. But in many cases where you need to do tomography, you might get lucky and be able to define the refinement area such that your peak is then significant in that area. But I guess in most cases that won't work. But you can do your initial tomogram reconstruction focused on a very local area in order to remove the problems of having an object which is not necessarily rigid through your tilt series. And then you can do your final reconstruction from your original tilts if you want to remove interpolations. You can deal with most of the problems, I think.
So just a continuation of this question. So what was the best resolution achieved so far? Like,
I think as far as the best published resolution is the structure there. Yeah. And we've gone significantly further, but our structures are moving at the moment rapidly, which is why it's not showing you. But certainly we can go past 6.
Since you're looking at single particles genuinely, what can you say about the symmetry of those particles? Do they have as high a symmetry as some of the icosahedral ones, or are they generally full of defects in the packing of the pieces?
Yeah. So the examples I've shown you today, the COP1 and the HIV, both mean there is no point group symmetry of the overall structure. Okay. You have local symmetries, and you have defects that are which are completely non-symmetrical random defects in some cases to accommodate the curvature in the lattice, basically.
Okay, and is there any pattern when you look at a few different particles? Do you see the same, some more symmetric features and some less symmetric features and so forth?
So in the case of HIV, essentially in the immature HIV there is a hexameric building block which forms a hexameric lattice, and as you curve that lattice at some point you essentially break that symmetry and end up into large defects. In the case of COP1, where you have this threefold structure, that threefold structure comes together in a number of different ways, and we can then go and identify those positions based on geometry and reconstruct those, and you see protein flexibility and different interactions between the proteins at different positions. But even with a subset of defined ways of interacting, you still in those vesicles have somewhere a more substantial defect which removes the need to close into some kind of symmetrical structure.
Might be the budding point or something.
It is the budding point in the sense that if we reconstruct buds, there is only one defect and it's at the neck. Yeah.
So with the working distances that you're achieving with the Cryo-CLEM, are you now in a position to start thinking about doing a super resolution optical?
Yeah, Kai, no, is it here? It's Kai here. Kai's been doing some really nice super resolution stuff, and yes, absolutely, that's a project which is ongoing in the lab. These are not great objectives. These are not water or oil immersion objectives at this stage, where there are people working on immersion objectives for cryo, and their optics are not bad, but they're not, let's face it, cooling objectives. You need to design a special objective, and that's a huge investment companies haven't yet made at this high level. So there are some problems there, but that's definitely a direction which people are going in, including my lab, and which will work, and where there are also potentially interesting benefits, because the fluorophore properties are very different at low temperature in some cases.
Okay, so let's move on to the next talk. So Ardan Patwardhan from the EBI is going to talk to us about all things EMDB.
I'm going to tell you about EMDB and EMPIAR and developments and initiatives surrounding these two archives. And I'll start by giving you a brief overview of the status of the EMDB, and then go through some of the... I'll then go through some of the services that we have for searching, visualizing, and analyzing EMDB data. We have new EMDB entry pages, which I'll show you. I'll show you a little bit about the EMPIAR, which is the electron microscopy pilot image archive. how to access EMDB data programmatically, and then tell you a little bit about our developments in the pipeline and our longer term plans and community interaction. So EMDB, for those of you who don't know, stands for Electron Microscopy Data Bank, and the archive was founded at EBI in 2002, and it's been run as a part of the EMD Data Bank project, which is funded by NIH since 2007. In terms of what we hold, most of it is single particle maps, followed by subtomogram averaging maps, helical reconstructions, tomograms, and electron crystallography. If you look at the number of maps that we hold, we are soon approaching about 3,000 maps, and in terms of the number of maps released per year, so last year we had about 600 maps, and this year we are already up to about 200 maps released. So something that we have seen happening during the past couple of years is what's become known in the electron microscopy field as the resolution revolution, and that's quite perceptible amongst depositions. So if you look at the number of released EM single particle maps and better than 4 angstroms resolution, this was relatively low for a very long time until about 2013. And I guess with the advent of the direct electron detector, we've seen a big jump now during the past 2 years. And it's quite interesting when you look at this in terms of the graph on the right here, which shows you 3 different bins of resolution. So the red one is for better than 4 angstroms, the green one is between 4 and 8 angstroms, and the purple is between 8 and 12 angstroms. And if you look at 2011 and 2012, you can see that you had more, they follow a normal trend where the low resolution bins have got more entries being deposited. In the last 3 years, you see that there's been a complete shift in the distribution. So we have a lot more entries in the 4 to 8 angstrom compared to the 8 to 12 angstrom. And this year, in particular, you can see that even the better than 4 angstrom resolution bin has got as many as the 8 to 12 angstrom. So going through some of the services that we provide in terms of searching, visualizing, and analyzing EMDB. If you want to search the EMDB, the best thing to do is to go to our website, and the link is provided here, pdb.org/EMDB. And there are several different ways that you can search data in the EMDB. One option is that on this page you've got these images. These are essentially quick access links to different basic categories. So for example, ribosomes, helicases, and so on, HIV. If you click on these images, you go to a page where you get the entries for these different categories. But from the left menu, you also have options to browse the whole archive, and you can also have a search option to search to perform an advanced search. So this is just an example. So if you were to browse the archive, you are taken to a page which looks like this, where you have filters which allow you to quickly drill down to specific categories, for example, based on EM method, authors, and, for example, resolution. We also provide the EM stats service, which is gives you dynamic charts which have been generated on the current state of the archive. So these are charts on many different aspects of the data. For example, you can see basic map release trends, resolution trends, publications, so which journals people are publishing in, and so on. For visualization, we currently offer 2 options. One is that we have a 3D viewer for all entries. So if you go to, for example, pdb.org/the entry ID/volume, you'll be taken to the 3D viewer, which allows you to interactively rotate and translate the 3D volume, and it also allows you to overlay the models on the volume. For tomograms, we currently offer a slice view, which allows you to look at the tomograms section by section, and that is shown on the right here. We've also developed a set of pages known as visual analysis pages, which provide basic analysis or basic sanity checking information on different entries. So I'm going to go through some of the different aspects of the visual analysis pages, and the easy way to access these pages is by the pdb.org/the entry ID/analysis. So one thing that we provide on these pages are these different images. So for each entry you have orthogonal projections in X, Y, and Z. We provide central slices now, and we also provide orthogonal surface views. So these surface views are generated at the contour level that have been provided by the authors. So one thing that we added last year was these images of the central sections, and this was a discussion that we had with James Conway and Sarah Butcher. And essentially the reason for doing this was that these central sections, so I should also mention that if you click on any of these images, you get a zoomed up image of these at the full resolution. And we essentially give you it's an easy way to do a quick assessment of the map quality. You can see how well, for example, capsids are defined. We also provide these different graphs. So it's the first on the left is the map density distribution, which can be quite useful, for example, to assess what type of masking has been done. The graph in the middle is the volume estimate, which is quite useful to look at whether the contour level gives you a volume which makes sense in terms of the biology of the specimen. And if people have done Fourier shell correlation and provided us with Fourier shell correlation curves, then we show these curves as well. We also calculate the rotationally average power spectra and show these. And the rotationally, so here are 4 different examples of rotationally average power spectra that I'm showing, going from, on the left, relatively low resolution to relatively high resolution on the right. And in the rotationally average power spectra, the Y scale here is on the log axis. And for the curves on the left, you see something which you typically would expect in the past from rotationally average power spectra, where you have a decaying function. In more recent reconstructions, what you quite often see is that people are using flattening of the spectra in the high resolution reconstructions, and you can see that the rotationally average spectra does not fall off but flattens at high resolutions, except for these cutoffs at some specified spatial frequency. What's also quite interesting with these is that you can see these so you can now see that for these high resolution reconstructions, you can see these bumps. So these are characteristic bumps for different types of secondary structure elements which are present here. So you see one here at about 4.5 angstroms for this virus, and for this ribosome, you can kind of see that there's a bump coming in here as well. If a model has been fitted to the map, we also calculate how well the model fits into the map, and we do this in 2 ways. So one is an atom inclusion by residue at the contour level provided. So here you can see the different residue ranges, and what we have here is essentially a color ramp scale going from green, meaning that the model fits well, the residue fits well into the density, to red, meaning that it's outside the map. So this allows you to look at find regions of the model that fit well or don't fit well into the map on a residue by residue basis. And here in the chart on the right, you essentially see for the whole model how well the atom inclusion is by if you vary the contour level. And you have two curves here. The green one is if you take all the atoms into account, and the blue curve is if you only look at the backbone atoms. So the visual analysis pages we calculate for all entries in the EMDB archive, but we also provide validation servers that people can use to perform validation on their own structures. And we're doing this for a number of reasons. One is it's a convenience for people to have these access to these servers, but it also helps promote use and reporting of validation information. And it's also been a good way for us to work with software developers to create standards for capturing validation data. So along these lines, we have set up essentially two servers. One is the Fourier shell correlation server, which allows you to upload two maps and it calculates the Fourier shell correlation. And the Fourier shell correlation curve that's calculated can be downloaded and then deposited to EMDB. Some of the features of this server is that it'll automatically calculate different thresholds which are in use in the EM field. And we've also talked to the developers of VSOP, Reliant, and EMAM2, and they now incorporate functionality to generate these FSC curves directly, so you don't have to use that FSC server, but you can directly from these packages generate FSC curves which can be uploaded to EMDB. We've also worked with Peter Rosenthal and Sebastian Wasilewski in his lab to develop, or actually to port the field pair validation server which he had set up to EBI. And this is now up and running, but what we still haven't done is to develop the deposition of field pair data. So you can use the field pair validation server, but this data cannot be uploaded yet to EMDB. And some of the requested features that people from the community have requested are, for example, that they would like to be able to read they would like the server to read other file formats and other angle formats as well. So this just shows you how the tilt pair validation, the results of the tilt pair validation server look. So on the upper panel shows you the summary results, and you also have per particle results as shown below. And the tilt angle distribution chart is interactive, so you click on a point there, then it will automatically take you to the particle plot below. So this was things we have had for a while, but quite recently, last month, we also introduced new EMDB entry pages. So the look and feel of the EMDB entry pages have been updated, and this is an ongoing initiative at PDB to try and integrate the way that we present both EMDB and PDB data. So if you looked at the new PDB pages, you'll see that the look and feel is quite similar. So this is the EMDB entry, and then if there's an EM PDB model, you can see that the look and feel of these is again quite similar. We've also now introduced an API which will allow you to have programmatic access to EMDB data. So this is part of the API framework, which can be found at pdb.org/API, and it's the main access mechanism for EMDB metadata for all our internal services, and we're also providing it for external use to make it easier. So rather than having to download the XML files and then pass XML files to get information, you can use the API to directly get a JSON which you can use in your programs. I should also mention that this API, again, we have integrated the API for EMDB and PDB. So if you go to API here, to the link, you essentially come to a page where you have not only how to access EMDB information, but also how to access PDB information and other related services. So moving on to the EMPIAR. So EMPIAR is the electron microscopy pilot image archive. And so what is EMPIAR? So EMPIAR is a 2D image database that we set up, which contains 2D raw image data related to EMDB entries. And it provides data for methods development, training, and validation. And since we started it up, we now have 25 released entries, out of which 2 entries that have been released are at more than 2 terabytes. 2 entries which have more than 1 terabyte. It's been cited in at least 7 publications, and as I'll mention to you later, it's going to hold the raw data which is related to the EM data bank map validation challenge. We are also looking, so right now entries in EMPIAR have to be related to EMDB entries, but we're looking at ways to expand EMPIAR to also accommodate other imaging modalities like FIB-SEM data. So in terms of functionality that we have implemented, so we have now EMPIAR entry pages. So each EMPIAR entry has its own page. In terms of uploading and downloading data to us, so we're talking here about data sets, raw image data sets, which can easily go into terabytes. So FTP has not worked that well for this, and we use 2 technologies. One is Aspera and the other is Globus. Both of these involve a server-side cost, but that is borne by EBR, so it's free on the point of clients to use it. From the client's perspective, the inconvenience is usually that you have to download a plugin, or you have to download some software to use this, but there's no cost involved on that side. We have also implemented functionality on these pages to make it easier for you to browse files and directories to download. So if you don't want to download the whole 1 terabyte data set, you can download individual files, but often even the individual files here are 2 gigabytes in size. And we also now assign DOIs, document identifiers, to each EMPIAR entry. So going over to some of the developments that we have in our pipeline. I think this is probably very important for the community. So we're working on an integrated PDB and EMDB deposition system, and this will go live sometime this year. So you've already seen the updated, those of you who deposited PDB have seen the new PDB deposition system. So this new deposition system will also incorporate EMDB deposition. And we are now at the stage that we are doing internal testing of the system. So when this new system goes live, as with is currently done on the XJ side, there'll also be an EM validation report which will be generated and sent to the depositor. So when you deposit, there'll be a validation report. And this validation report is sometimes asked for by your referees and so on to have a look at. So the initial focus that we have is to generate EM model validation. So this is what we have. So on the model side, we can essentially incorporate more or less the same types of things that are available on the X-ray model side. On the EM map side, what we initially have is very bare bones. It's essentially just a table of experimental information. So it'll just be basic minimal information about the entry. In terms of what type of components that we will include in the validation reports, there's the EM validation task force which will advise us on how to proceed. So on EMPIAR, we have also now implemented a way to visualize the data in EMPIAR on the web. So this is challenging, keeping in mind that we have terabyte-sized data sets, but we're using Omero, which is a sort of client-server system, to do this. And we have a functioning prototype. It works for many file formats like MRC and so on, but it's very dependent on which formats are available in the bioformats, which is a library which is used by Omero. So I showed you the slice view, which was used for looking at slices of tomograms, and we are working on improving the functionality for looking at slice data. And the first step that we have taken for this is to develop what we call the volume browser, which essentially allows you to now not only look at slices in the Z direction, but you can instantaneously switch between looking at any three orthogonal directions and looking at the slices in between them. And we will be introducing this not only for tomograms, but also for all entries in the EMDB. We also have a project in progress with, which is a collaboration with Martin Wynn at STFC, which is the PDB project. And the idea with the PDB project is to search for matches in EMDB and PDB based on structure, on shape, rather than simply on metadata. So, and that's one use case. The other is to actually just align structures, because quite often we have related structures but not aligned in these databases. So the basic use case that we were contemplating was that you could upload a map to our servers, and then we had a shape database against which you could match your uploaded structure, get some potential hits, and then we would then do shifting and rotating to do a finer alignment, and then you would have the best aligned matches. So this is quite a challenging project, and what we have initially done is to look at matching structures which are already in PDB and EMDB, and our initial use case focus has been on ribosomes, because rather than having 100,000 structures, we limit ourselves to 1,000 entries, but this still means that we have about 1 million pairwise alignments to do. So this is just what we have in advanced development right now. So we have a server where you can select, for example, one entry, and it will give you the top hits against that entry. And as you can see, the top hit is the entry itself, and this is because it actually does an alignment against it itself, and it does not necessarily come out with a score of 1.0. But if you look at, for example, the alignment against the second best match, which is 5788, we have interactive visualization of the alignment of these two structures. So you have the 5787, so these are two ribosomes. 5788 is one which is supposed to not have the L16 protein in it. So you can rapidly visualize it here, but we understand that for a lot of advanced work, people are not going to want to do it on the web, but they would want to download, for example, the alignments and then perhaps play with this in Chimera. And we have this download pose button, which essentially will, when you click on that, you can download a Chimera script, which will allow you to load these alignments into Chimera to play with it. And this allows you to do things like this. So in Chimera, you can then do difference. You could rapidly do a difference map between the two structures, and this is what you can see here. So the difference is shown in line in green here, and so just overlaying on the left is a picture of the ribosome with some of the proteins. So the L16 protein is supposed to be here, which would correspond to this. But you can also see that there are other differences that are seen here. So a lot of this might be because of the way that so doing difference mapping is not trivial, so there might be issues with that. But what I just want to illustrate was that now we have a way to rapidly look at these types of things. So if you look at some of our longer term initiatives, so I showed you the volume browser, and we want to go a step further with the volume browser, and that is to also integrate, to start incorporating segmentation data into EMDB, and that will allow us to sort of take it a step further, where we can integrate tomographic data all the way down to molecular data. And then finally, in terms of community interactions, I wanted to mention that a lot of the things that we do right now are, we want to drive them in terms of testing them on the community. So we're trying to do continuous testing from early stages to of development, and we have several different services that we need testing on. So we would be very happy to have more volunteers who would volunteer for testing, and we are happy to visit other people's labs to perform this testing. We also want more data, so people have started to deposit a lot more Fourier shell correlation curves, but again, we would like to encourage people to do this with more. Again, deposition of raw data to EMPIAR. And for many of the initiatives that I've mentioned, we would also like to have test data. So for example, segmentation data and imaging data from related fields like 3D scanning, EM and soft X-ray tomography and CLEM, and also from, for example, small angle scattering. I'll just briefly go through this. So something that's might be interesting to highlight is that there are going to be, in the context of EM data bank, there are going to be two EM validation challenges. So one is the EM map validation challenge, and the other one is a model validation challenge. And at least for the EM map validation challenge has come so far as that they've decided to have about 7 comprehensive data sets in EMPIAR that people will then be able to download to reprocess. We also organize many different workshops with community experts to get identifying challenges and gaining community consensus. And 2 of the more recent ones, one was the data management challenges, and the other one was a cellular context for the macromolecular world in 2012. And I just want to mention this because we are also planning to organize one later this year. And with that, I just want to acknowledge all the people who are involved in the EM Data Bank work and all our partners. So the EM Data Bank team is quite big. I just want to especially recognize some of the people who are doing this work, including Eduardo, Ingvar, Jose Salavat, and Andrei. And then at Baylor, Vachu and Steve are partners in EM Databank, and we have RCSP working with us on EMDB. Jason has been a partner, especially when it comes to Omero. Martin Wan and Agnel at STFC with both CCP-EM as well as the PDB shape. And at Birkbeck, Maya Top and Irena have been very helpful again in the development of the shape matching server. With EMBL Hamburg, Dimitri and Anne have been working with us on SACS. Peter Rosenthal and Sebastian on the tilt pair validation server. And then I would also like to thank many of you and all people who are depositing data to us and also who have volunteered already to act as testers. And then finally to our funding agencies.